I'm sure some more people will be logging on in the next little bit, but it's my pleasure to introduce Dr. Andrew Weng, who's our speaker today. Andrew is a professor in our department at UBC and also a PI in the Terry Fox lab at BC Cancer and a service hemopathologist at BC Cancer Agency. He's also serves as a director of the clinical flow cytometry laboratory at the BC Cancer Agency as well. His research focuses on the pathogenesis of human leukemia and lymphoma with specific interests in T cell, acute lymphoblastic leukemia, and B cell lymphomas. His lab seeks to uncover mechanisms underlying the initiation and propagation of these diseases using de novo experimental models derived from human primary hemopoietic progenitors, which may reflect the biology of human diseases more accurately than cell lines and mouse models. His lab's experimental approaches rely heavily on primary cell culture, viral-mediated gene transfer, CRISPR-mediated gene editing, RNA-seq, ChIP-seq, ATAC-seq, and flow cytometry. These efforts are programmatic and embedded within TFRI-funded program project teams in leukemia and lymphoma, respectively. His work has been widely published, and we look forward to hearing more about it. So thank you very much, Andrew. Turn it over to you.
Thanks, Will. Let me see if I can do the share thing we talked about.
Let's hope it works.
So I believe I'm sharing.
Yeah, we see your introduction slide.
Presentation. Okay, so hopefully I'll make it through quickly and leave plenty of time for any questions or discussion. But I'll be talking today about some kind of basic research discovery work that we've been doing using high parameter cytometry, either mass cytometry, CyTOF, or just regular conventional but high parameter flow to answer and understand the biology of human B cell lymphomas based on primary patient material. And then the second half of the talk, I'm going to then talk about how we can use the things we discover from that sort of stuff to inform and improve the sorts of clinical assays that we can deliver to the patients of BC. So I thought that clicking would help. Okay, so I think I'm on the second slide outline. So first, I'll just give a historical background of the flow capacity, both in the research and the clinical sides. Then I'll kind of make an argument to you that higher parameter analysis is always better. I'll give then kind of a brief overview of two different research projects that we've done to try and answer basic biological questions about lymphoma pathogenesis in primary patient material. And then finally, I'll close out with what we're doing and what we can do to have that technology and that knowledge trickle down to and improve the way we deliver care clinically. So here's a quick long-term overview over the last 20 years of flow cytometry as it's manifested here at our institution. So I think you can see my mouse. If not, just someone yell at me. With technology improvements and dye chemistries, there's been this consistent march towards higher and higher parameter analysis. And the blue line here is the blue lines are what we've been able to do in a research setting. And you can see it's risen substantially over time. We're generally about 40 parameters, 50 parameters of capacity. And the research or the clinical lab has always, by definition, has kind of lagged a bit behind, but we've been doing our best really to chase the capacity and really keep our clinical assays up to date and using the latest technological improvements. One question is, well, why? You could say, well, we were diagnosing CLL with 3 colors fine 20 years ago. Why do I want more data than that? But it's a matter of improving the data and also improving the efficiency of the assay, especially as biopsies and clinical material becomes more and more precious and people are less likely to go carve out a big chunk of tissue these days. So with more parameters, you get more information from each cell. You get a much more broad picture if you can look at all the parameters and see the entire picture at once rather than small snippets of the information at a time. By definition, you can resolve populations directly and prove that the cells are different from one another rather than just inferring that they're different based on abundances across different tubes. It's a more efficient use of biopsy material. As I mentioned, biopsies are getting smaller and smaller, and there's nothing we can do about that. And lastly, kind of as a joke, it's 2025. Like, why we live in the technological realm of 20 years ago? That's why not? So, and this is a simplistic little graphic to just show how much information you get when you add on more and more parameters, and you get much more resolved information. You can discriminate things from one another, and you just get a much more informative view on what's going on. So one aspect about gathering more data with more parameters is, and this is something people sometimes underappreciate, is you really do have to update the way you analyze this data, because with conventional, for those of you who do flow, basically, no matter how many parameters you have, the conventional way is to draw a gate around a two-dimensional view of that data, looking at only two parameters at a time. And then within those gates, look at a different two parameters, within those two, a different two parameters. So it's called serial bivariate gating. And the more colors you get, exponentially, you get, well, it's not exponentially, according to this mathematical expression, you get more and more and more and more plots. And at some point, you can't possibly absorb that information or organize it in a reasonable or interpretable way. So that's led to kind of a more holistic way of looking at the data and looking at natively in all the dimensions at once. Now, you can't even perceive that. And, we're talking about creating this, for example, we're talking about analyzing data in 32 dimensions. You can't even imagine what that looks like. The best we can do seemingly is 3 dimensions. But this is kind of, if you extrapolate this up, another 29 dimensions, you get a sense for how complex the information space can be. But also, even though we can computationally do that analysis in K dimensions, if you will, then we still have to look at it as humans. And so that's one reason why you may have heard about these different mapping techniques, UMAP and TISNE. Basically, it's taking all those 32 dimensions, for example, and squashing it in a particular two-dimensional projection. Now, you lose information by definition, but that's one of the goals of the different algorithms, is to try and show as much variation or capture as much of the greatest parameters of variation in the data, and show relationships, so they might not be linear, but nonetheless show relationships between the different populations. So this is perhaps a 2D squash, this picture here, a 2D squash of up to 32 dimensions of data, at least in this data set. So now I'll move a bit into, with this new sort of technology, what sorts of questions we've been trying to address, kind of to understand the biology of lymphoma more so, which is a complement to mechanistic based, for example, animal studies where you manipulate a system, you create isogenic comparisons, you do very regimented controls. That kind of focused investigation is in large part motivated by observations that are made in primary patient material. And so that gives us, by looking at the primary patient material in more and more depth, you get more and more a view of like, well, this is really what's happening in the disease. And then you can ask questions like, well, what is driving that variation, or what does that variation mean? Do we even care? Does it have any clinical meaning to it? Or are we just kind of creating more and more pigeonholes to divide things into that we don't really know why we're doing it, but we do it because we can? So hopefully I'll convince you that there are reasons and benefits to doing this sorts of dissection of analysis. So our generally stated objective in these 2 studies that I'm going to kind of go over are to identify relevant inter and intra-tumoral heterogeneity in human B-cell lymphomas using high parameter cytometric methods. So as I kind of alluded, many of you are familiar with conventional flow cytometry. Routine, almost every lab can do it. Mass cytometry it looked like it was going to be the wave of the future. I have to say that it's kind of receding as a useful technique, but it had its day where it allowed more parameter analysis than competing methods. But quickly, as you may be aware, technological advancements and vendors have really upped their game on the conventional side, and now you can pretty much match the performance more or less that mass cytometry was offering with conventional laser-based fluorescent fluorochrome mediated analyses. And the analysis is better, for example, using spectral approaches rather than dedicated channels. So these are the kinds of technologies that we're using. I don't think anyone would ever see mass cytometry implemented clinically, but we can actually, in our now BCCA, we're implementing spectral flow, or can implement spectral flow if the data needs it clinically at BCCA. So the samples we're looking at. So these are single cell suspensions from diagnostic lymph node biopsies. So many of the tissues that of lymph node biopsies that get excised in the province get sent into BC Cancer for analysis, which serves two missions. One is getting a useful diagnosis, and the second mission is we actually do bank material, and then once it's consented for your research use, then we can use it to discover more about the disease rather than just kind of keeping the status quo with diagnosis. We're looking to discover more and hopefully bring an added value there. So two of the most common nodal lymphomas that you'll out there are follicular lymphoma and diffuse large B-cell lymphoma. So these two studies utilize that because we have most samples and we can actually try and draw more generalizable conclusions as opposed to a small number of anecdotal reports, for example. Plenty of normal reactive control, so we're able to put these in the context of normal biology. So this work I'm going to start off with was done, spearheaded by Shuhai Wang. He was a postdoc in my lab at the time, but now thankfully he's decided to stay with us and really takes care of the technical aspect and the informatics aspect, having our clinical operation run. But he remains involved in our ongoing research studies. So, and you'll see some of that ongoing work in a second. But anyway, so you can't really do a clinical study without lots of patients. So, and you need a lot of material. So you kind of need a central place where that material is captured in order to really get enough, critical mass to actually find information from the variation that happens in the scope of patient samples. So here we are able to collate 155 follicular lymphomas, and we have 36 reactive lymph node controls to compare them against. We took cryopreserved tissue suspensions, we thawed them, or Shuhai thawed them all, and did 40 parameter cytoph. We looked at B cells and T cells, so those are the most important or relevant populations, at least, that we can access by cytometry. Just to mention, we did a bunch of other, collected a bunch of other data to inform on this, both clinical information, which was provided by the clinical lymphoma database that's been built originally through Joe Connors, but now maintained through David Scott and all our clinical colleagues on that side. But we've also kind of supplemented that kind of descriptive work with as much modern next generation sequencing based characterization of both DNA variants and as well expression RNA-seq based expression profiles to really kind of give some depth and more orthogonal information to help us understand what's going on with the admittedly relatively lower parameter cytometric assays. Because although 40 might sound like a lot, the transcriptome's got 20,000 transcripts. So, but not all of that's informative. So we'll see what we discover. Before I go into the details of that one, I wanted to point out that I'm also going to tell you a quick story about a similar study using diffuse large B-cell lymphoma instead. This was spearheaded by Michael Nissen, a former postdoc in the lab, but also Shuhai and Michael kind of worked as a team to get these two studies off the ground and to where they are now. So we did a small discovery set in Cytometry, but we soon discovered that we didn't really need Cytometry to answer the sorts of questions we wanted to. And in fact, in order to look at the signals that I think we were seeing in the Discovery 1 cohort, we needed a lot more patients. And that became a little bit arduous because because some of the material we needed, the material wasn't frozen as carefully. And so we needed kind of a more higher data capture modality. So we went back to conventional flow, but at the time the Symphony had just come out. And so we were able to capture 23 of those 40 parameters with conventional flow. And we felt after the initial view of data that we could actually get the answers we wanted to. So we narrowed it on a T-cell question here in 23 parameters. We enlarged the discovery set. I'll show you what we discovered. But for clinical application, you may well know you can discover something, but no one really wants to touch it unless you can externally validate it in an independent cohort. So we actually, because the nature of the data and the findings we had in the Discovery 2 cohort, we then went ahead and we actually used existing flow cytometry data. Like this was already in the computer. Because we had been running 14 color cytometry routinely clinically, we actually had enough information to just simply ask retrospectively in the data that's already come in the door for routine diagnosis, can we look at the features that we discovered in the high parameter platforms and see, can we validate the predictions, the clinical predictions in actual clinical data? And I'll point out to you that I think we did. And so just to point out, we use cryopreserved banked material for the discovery phases, but the validation data was just what was coming in the door, fresh tissue, solid suspensions, which were analyzed as a course of our normal routine characterization for LPD panel, lymphoproliferative disease panel, clinical testing. And so we, again, merged various other data forms. We've got some DNA characterization, and in this case, some functional studies. So I'll point out those to you as well when we get there. So this is just an example to say how many data forms, and there's a fair bit of managing many different modalities and kind of connecting it all together. It's a fair bit of logistic work, but I wanted to point out that this there's a lot of work behind what I'm about to show you. So collecting the data is one thing. And believe it or not, we've collected all that data. The data collection went pretty quick. Organizing it took a little longer, but by far and away, the largest effort was analyzing it and looking for patterns. So part of that was hindered by the fact that traditional grading hierarchies, this is how we initially started with the ideas. This is how we all, at least clinically and many manually, people look at data in this fashion. You have a series of bivariate gates, like I mentioned. There are these hierarchies. It's kind of hard to kind of put it all together because you're looking at the data in such fragmented views. It's biased because you have to pick which path of gating you want to pursue, and it's inefficient, and then you're unreproducible because everyone does it differently, and is actually relatively slow. And so we quickly realized that we weren't going to make any progress on this data if this is the approach we were going to have to use. So in comes, again, I mentioned multi-dimensional clustering. It's agnostic of any preconceived notions you have about the utility of one marker over another. It just looks at all the data all at once. It's unbiased. It is subject to random effects because of the way the data is analyzed, and it can be variably efficient depending on how the code was written or what assumptions were made in the particular algorithm you chose. But one thing about that is once you come up with your decided approach and you've had talented programmers rewrite the code to make it more efficient, this is scalable. That is, once you've chosen it, the computer just goes and does it. You don't need to then say, oh, give me another 4 grad students to spend 6 months gating the data in unreproducible fashions. Anyway, so there are different ways of clustering, and I only throw these terms out here in case some of you are familiar with these clustering algorithms. They all have different pros and cons, but again, it's kind of, you have to choose the algorithm to what your data question is and look at the results of it. And do you like it? Does it tell you something biologically or not? Anyways, this is just to also mention that, I wanted to give you a visual to emphasize this issue of, and there's only 3 dimensions. Remember, there's K dimensions we're talking about, or 40 dimensions in some of the case. how you rotate or squash the data to really change the two-dimensional depiction of how you can view, oddly enough, it's a duck with orange feet, or if you rotate that, you get different ways of cutting that duck into pieces, and so you get more information. Anyway, so because you actually thought this was his foot, but actually now it's just a disembodied foot floating in another dimension. Anyway, so now we're looking at data. This is actually this is a two-dimensional squash of 40-dimensional data from the data that Shuhai collected on 151 folliculars and 36 reactive lymph nodes. This is what reactive lymph node data looks like in 40 dimensions, just the B cells. And this is what the follicular lymphoma samples look like. I point out to you that, well, then we use phenograph clustering. Each of the dots is colored by which patient it came from. So every color is a different patient, and the patient's cells intermix quite intimately here. And sometimes this area here are composed of cells from only one patient. No other patient has cells in that position. So one thing we routinely do is we cluster it just to find group things, like things together. And this is one particular clustering approach. And now we see that we kind of simplify the map. And we say, here's a type of cell. It can come from many different patients, but here's a type of cell. And then we can use our knowledge just to put it in the context of how we understand B cell development and label them as naive B cells, memory B cells, in this case, pre and post class switching, germinal center B cells, plasma cells, etc. But because now we know, and there's a utility of having normal controls, now you know what the spectrum and the scope of normal B cell progression is, you can find those cells. They're still present, they're still persistent in residual tissue of the tumor-involved node, but we can find them. And that helps us use to focus in on the abnormal cells. And interestingly, now this is quite clear, where are the abnormal cells? Well, they're quite clearly identified in multi-dimensional space. One thing I wanted to point out is, it looks like that patient, or sorry, I mentioned that this cluster here is coming from a single patient. And if you like the notion that every patient's tumor is different, sure, that can fit with you. However, you also know that there's two areas here where cells from many different patients, I'll have to go back to this one, lots of different patients plot cells into one of these two areas of the phenotypic map. And so you can measure that degree of intermixing of patient cells by entropy. And basically, high entropy means that cells in that region of the plot are occupied by many cells from many different patients. So the more different patients that plot into that region, the higher the score goes. And so this is a very high score in normal cells because everyone's normal cells actually look like everyone else's normal cells. But when you have a very low entropy score, when this is like kind of a private or a unique phenotype that only one patient holds. But as I pointed out, these two areas here, it's kind of intermediate, which means lots of different patients actually are plotting their cells in these two regions. You might say, well, that's follicular lymphoma. They all have follicular lymphoma, but guess what? There's two. We didn't think there'd be two. I don't know how many we thought there'd be, but there's not just one type of follicular lymphoma. There seems to be two, at least two. So we kind of highlighted these two areas into the red and the blue mark. And we, I'm going to cut to the chase. And basically, we determined that we believe there are two different types of follicular lymphoma, not one, but two. We call them type A, which is the kind of one that we thought we'd find. They're very much like germinal center B cells. They overlap in phenotypic space with normal germinal center B cells. And so we all think follicular lymphomas derive from germinal center B cells. Well, this is what we're usually thinking, what we're talking about. And that does comprise about 28% of all tumors in our cohort. Well, what's this other type? So there's another type, makes up 18% in this cohort of samples, which they don't look like germinal center B cells. They are closer to memory B cells. So we call those memory type follicular lymphoma. You might say, well, who cares? It's just a different flavor. It's all follicular lymphoma. But here's the reason why it's different is if you look at the risk of transformation. So many of you are familiar that follicular lymphoma is a low-grade lymphoma. That is, patients can just have a lump and walk around with it for years, and if it doesn't do anything, they're all good. They might not even need any therapy at all. However, there's a 2 to 3% chance per year that that low-grade lymphoma will transform. Something will go wrong, and that cell will blow up into a more aggressive lymphoma, often diffuse large B-cell lymphoma, but that's the most common. And that clinical event requires chemotherapy. Now you have to go see, now you have to get CHOP times 6. Now you have to go see the doctor. Now you have to get involved. Now you have to be sick. So how can we predict whether or not the patient's not going to need anything, or are they going to they're going to be worried about in the next couple of years, they very well could transform and need chemotherapy. And this is the risk of transformation, the risk that clinical event, that transformation event, that you're going to need chemotherapy is going to happen. So the risk of transformation is statistically higher if you're of this type B, this alternate memory-like follicular lymphoma, than the kind of the run-of-the-mill germinal center type A type lymphoma. So I would argue too that it's bad news if you get labeled with a memory type follicular lymphoma. You might need to be followed more carefully by your physician, and if we ever understand about the biology of it, maybe we'll actually do something proactively to prevent that transformation event. But otherwise, the rest of lymphoma, it's a pretty indolent disease. You're not necessarily going to transform, or at least a high risk of it in your lifetime. And this is the findings this we published in Nature Comms in 2022. I wanted to point out that others have followed up on this. And although we only had clinical information on risk of transformation, kind of a gold standard in clinical world as well, did the patient live any longer? I don't care if they transformed or not. I don't care if they had to get intensive chemotherapy. I don't care if they're sick and puking and bald. Did they live or did they die? That's kind of how clinical realm goes. And so here's another group. They actually use RNA-seq, and they came up with a larger cohort. They came up with 46 clusters. They lasered it on 2, but in the end, they found that their 2 clusters they found were basically identical or highly overlapping with what Shuhai had already defined as the germinal center type B-cell lymphoma and the memory type B-cell lymphoma. And here they're showing, now they had enough patients, enough clinical follow-up that they are actually able to score survival as an outcome, where you live or die. And so you can see here that indeed, just like we said, the memory type was a high risk for transformation, they're showing that the memory type has a lower progression-free survival. So that just came out a couple months ago, but I think it's important to note that this concept is not just, well, we saw it and no one else did. It's actually been validated and has a meaningful clinical outcome at the level of survival. They came up with an IHC algorithm to kind of get away from that. I don't really like that idea, but that's what we do to get things published. But what does this all mean? What does it mean biologically for how lymphomas progress? And so I just want to give you a couple files. We collaborated with Ari Melnick's lab to realize that ARID1A mutations, which aren't the most common mutation for lymphoma, but they're there. I think it's under 10%, but basically ARID1A mutations are enriched in this memory type. And in their mouse models, they actually showed that ARID1A mutations lock cells into a memory state. So this all kind of fits pretty well. We found and these ARID1A mutations have an increased risk of transformation. So it kind of all fits together, but now we're getting a view of what is the underlying biology of this memory type follicular lymphoma. It seems to be cells are trapped through various types of mutations. In this case, a chromatin modifier, ARID1A, a sweet SNF complex, that locks cells into the memory state. We don't exactly know yet why the memory state is so likely to transform. We have projects going on to look at that, but now I think we have a bit of a genetic handle on what underlies this phenotypic difference between memory and germinal center lymphomas. Another kind of correlate is another thing that Shuhai discovered by looking at a co-occurrence analysis of B cells and T cells, because we had both sets of data, is he found that TFH cells were significantly associated with the memory type, but we didn't find that same in the germinal center type. And if you are familiar with normal germinal center B cell biology, you're like, that doesn't make any sense. Follicular lymphomas need TFH cells. So why would germinal centers need TFH cells. So why shouldn't we be discovering TFH cells in the germinal center type, not in the memory type? But then if you put that together with, again, work from Ari Melnick's lab that BCC and not me directly collaborated with, but they realized that EZH2 mutated, which is one of the most common lymphomas, mutations in follicular lymphoma, mutations in EZH2 actually reduce follicular lymphoma cells' reliance on TFH cells. And instead, the cells kind of adopt this dependency on follicular dendritic cells. So they kind of switch over from needing TFH and instead needing follicular dendritic cells, so-called a light zone lymphoma, because there are more FDC cells in the light zone. But lo and behold, EZH2 mutations are significantly more abundant in our type A, the germinal center type, than our memory type, which kind of all fits together in that the GC types are EZH2 mutated. They don't need TFH cells as many as well. Now they need FDCs, but that's why we were actually, I think, detecting TFH association in the memory type. They still need TFH help, but type A, the GC type, doesn't need it. So I just wanted to point this out because sometimes even our assumptions about normal biology don't play out in the actual lymphomas, we think, if we're too simplistic about making the associations. So here's the overall model of that, is that we think there's two types of follicular lymphoma, different mutations associated with the two different types. They have different T and environmental interactions that drive them. That's a kind of, so I think that it was initial characterization of variation in clinical material, but if you put it together with other work, then you kind of realize that there are molecular, there are DNA variants that underlie these differences, and there are important environmental interactions that support these differential biologies. So we did the same thing. Michael Nissen, as I mentioned above, did almost the exact same study with diffuse large B-cell lymphoma. Whereas we saw this kind of these two sub-flavors of follicular in Shuhai's study, we didn't see that in DLBCL. Basically we saw is every patient's sample was different. You might say, well, what about ABC GCB for those of you familiar with that? We could see it if we parse the data that way, but it wasn't the sort of kind of close overlap that we saw in follicular. Basically, every patient's tumor was still phenotypically distinct, but kind of looser connected through ABC and GCB types. So instead of focusing on the B cells, we decided let's look closely at the T cells in this particular case. So this is when you have this many parameters, you can divide the cells into many more groupings rather than just T and B or various other subsets. The distribution differs between reactive lymph nodes and diffuse large B-cell lymphoma. So they have a overall different T cell composition. Maybe that's not so different. But what we did was we took each and every one of those clusters and we asked, because we had survival information, we asked, are any of those T cell clusters here, any of the different colored plots here, in a very agnostic way? We're not saying, oh, I think this or I think that, just like, tell me what's going on here. Different clusters, actually, some clusters have a low risk of dying, and some clusters have a high risk of dying. So if you just look at the heat map of the expression of markers in those good versus bad categories, you can come to the realization that one thing we saw was that the bad clusters, if you look at all the patterns of colors, this is now humans looking for interactions. So wow, they're all 57 positive in the bad prognosis categories and all 57 negative in the good prognosis categories. So we went with it. And just to recap on this issue of ABC GCB, this high CD57 positive subset of 8 cells, they're more abundant in ABC. So maybe that's part of it, but they're actually also present in GCB, and they're kind of dog's breakfast mixed up between different mutational types. So it's not just a recapitulation of cell of origin, ABC GCB distinction. It's not simply a re-reflection of mutational categories. It does seem to be kind of its own thing. But we proved that statistically. And so in our discovery cohort, in multivariate analysis cell of origin, we actually found that the CD57 class of CD8 cells predicts a significantly worse outcome in terms of disease-specific survival or time to transformation trends and progression-free survival. So it has clinical meaning even when you control in multivariate analysis for cell of origin. So it's not just an echo of cell of origin. It's its own thing statistically. It's also independent of IPI, which for those of you familiar with that, that's another thing that people like to look for. And this is the validation cohort. So just to point out that it actually did validate. But what are these 57 positive T cells? Without going too much into detail, because it doesn't, it takes more time than we have to go over. But basically, CD57 seems to be a marker of T cell senescence. And these cells are cell cycle arrested. That is, you're not getting any more of these cells. They're there, but they're not going to proliferate. So, So actually, we wanted to test that directly. That's what the cartoons say, but is that actually what's happening with the particular flavor of 57 positive T cells that we're collecting, seeing in these samples? So we took 6 DLBCLs. We sorted the cells into the CD857 negative fraction and the CD8 CD57 positive fraction. We stimulated them in vitro with CD3, CD28 beads to mimic TCR engagement and asked, did the cells proliferate or not? Well, we actually found exactly what the literature would have suggested, is that the 57 negative fraction, they proliferate when you stimulate them with a mimicking of antigen. If they're 57 positive, they don't. So I think it's the actual functional data from actual patient samples saying that these cells actually do behave differently. And this is just a correlative finding that if you look at the T cell diversity in the 57 negative set, the proliferative set, they're all they're quite diverse. But if you look in the same patient from the 57 positive fraction of those cells, they're generally less diverse. It's a small sampling, but I just wanted to show it to suggest that these cells that we're talking about, these senescent post-replicative CD57 T cells, I think they've actually seen antigen already. They've already responded to the tumor cell antigen load, the neoantigens that they're seeing. But guess what? They're not going to divide anymore. They're kind of like exhausted soldiers. They're not going to really do much more for you in an ongoing immune response. So is that why? I'm not sure, but I want to tell you everything I knew about these cells. So we had 2 different stories here. One, 2 different types of follicular. Second, that we can identify senescent like T cells, and they're predictive of poor outcome in DLBCL. It took us 40 markers to discover these findings. We think we can recapitulate it with 28 or only 3. So depends on what you find, how many markers you're going to need, but you do need many markers initially to discover them. So I'm going to finish out here in the next 5 to 10 minutes with just quickly going over, well, those are the biology stories. How do we translate that to daily clinical work? So to point out to you, test volumes, whether it's driven by medical patterns or just straight up population growth, I can tell you the clinical workload is going through the roof. We can't even keep up. And 15 years ago, we're triple the number of specimens. The exact same flow lab was handling 15 years ago. So we couldn't scale the old procedures. The only way to deal with this population explosion or test explosion, we need new approaches. So this is where technology comes to the rescue, I think. This is our old workflow about when I took over the flow lab some 20 years ago. There's a lot of manual steps, a lot of tubes handling, a lot of people doing a lot of pipetting. Probably the only automated thing we had was there was a carousel loader on the cytometer, but otherwise it was a complicated, you stain 6 tubes, you analyze it manually, humans analyze it. If you think that you have another thing, you go back because you didn't have enough parameters to ask all the questions in the first place. You go back and add a reflex add-on. Let's do 4 more tubes because we didn't really get enough information. We didn't run 10 in the first place because no one's going to run 10 tubes. Long, convoluted, heavy, a lot of repetitive tasks, humans doing this effort. So how do you fix the situation? Well, we went to 8 colors in 2009, and that dramatically decreased the number of tubes we had to stain. We didn't have this two cycles through to cover all the information. We could catch it in a single round. And that was what we did then. That doesn't solve all the problems, but that was what we did in 2009. Then in 2015, again, we're trying to chase more parameters. We got more data. We didn't actually decrease the workload, but we certainly got a whole lot more information by going from 8 to 15 colors in the clinical data. But then in 2020, what became our kind of bugaboo was, well, it wasn't bugaboo, but new automation comes online. Well, why not make our lives easier? Let's bring in some lab automation. So we got rid of one of the steps of the human pipetting step, and we have now a little robot to do that, and that helped quite a bit. Another thing that came up though was at this point in 2020 we were handling 4,500 samples a year, and that took an entire person of manually gating there all day long, 7.5 hours a day. They're just sitting there gating. And so that's not very scalable, and it was inefficient and not reproducible. So this is when Shuhai took a lot of the stuff he learned in analyzing CyTOF and high parameter data to, this is just a table to tell you these are all the reasons why machines are better than humans for this particular task. Fatigue, subjectivity, not scalable. Anyways, lots of reasons. So this is where Shuhai took all that he learned from kind of the multi-dimensional gating tools that we're using to analyze the CyTOF data, and he applied it to analyzing the clinical data, and that reduced what previous was a whole person gating all day long to now 1.5 hours of computer time per day. So that really did make a big difference in actually getting the work done. I don't think we would have, the lab would have continued as it is if we didn't have this improvement that Shuhai developed. This is just a quick view on that. Is multidimensional gating quickly separates the malignant B cells here from the normal B cells, two different flavors of, or very closely similar normal B cells here. It's like the computer just separates them for you. You don't have to sit there and find what made the cells different from each other. Multi-dimensional analysis kind of just drops in your lap. And then other informatics tools are just kind of like, you don't need people to sit there pushing gates around. Shuhai wrote routines to automatically gate them and display them and all the various. So this is how we look at the clinical data. But there's all this generated by a computer. Currently, it takes about 3 to 6 minutes to generate these with the data file, but it's a whole lot quicker than a human sitting there pushing gates around. Anyway, so getting back to what we're doing to make the clinical work better. After we kind of implemented the automated gating there, we are going to 32 colors. The panel's complete, and now we're just cross-validating before going live. We expect to go live in the next few months. With a 32 color clinical assay, again, you get a lot more information. The workflow is simplified. We're still doing automated gating. I'll show you what we're doing on that front too, but I also wanted to point out that there's still a lot of people pushing cells around. So this is where our newest little toy landed in the Flow Lab last week. It's a walk-away automation step. So you load the human loads the samples, the machine does it all, and then just at the end, you just load the cells onto the cytometer. So the goal here is we can't keep up with the work unless we kind of stay with the times. And so walk-away automation is really going to save that day. But then I wanted to quickly close out with telling you about, there's still improvements to be had in the gating structures. I just want to take you quickly through what we've done there. This is work that Ainsley Hill, a programmer in the lab, has been focusing on. Basically is how much do we really need, or how much of the analysis can we get the computer to do for us? And the plots I showed you before, basically we were offering up, well, here's some different B cells, here's how they look. In this case, we're actually trying to classify the B cells and tell you which are the abnormal ones and which aren't, rather than just giving you, here's population 1, 2, 3, 4, you make the call. So we manually annotated under 200 samples. These were clustered in multidimensional space, and then we collapsed the clusters based on manual annotations. So you could roughly see that all these clusters here are different flavors of B cells. There's not much resolution in the other populations because this is a B cell tube. So that's mostly the markers are dedicated to segregating the B cells, and they did. But you can also see here, just for those of you who are kind of clinically oriented, sure, the B cells are here. They're all identified as CD19. Here's some T cells are identified by CD3, but here's CD5. Okay, the T cells are CD5, but okay, there's a subset of B cells that are CD5. So those of you know, those are probably CLL cells. And what you then do is after you have this map, you take your new patient sample and you cluster it with that reference map. And then it says, well, your patient had cells here, 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 and here. And okay, that patient, sure, they had some granulocytes and T cells, but you have this flavor of B cells. They didn't have these other flavors, they have this flavor of B cells. So if then you know that that's, well, what flavor of B cell was it? Then you ask, well, what reference cluster did it pile on top of? In this case, it piled on top of the CLL cells. And this is that case, for example. So the patient's cells are yellow, the reference population of CLL cells are in blue. So what kind of B cells did this patient have? I'm going to tell you, it looks a lot like the 44 previous CLLs that we've seen in this data set. Sometimes those overlaps are high, they're almost superimposed. Sometimes they're kind of, a little bit denser on one edge, and sometimes they're close by, but not really even necessarily overlapping. So in order to use this data, not in addition to just telling you what it is, we're also telling you what their confidence that what is the level of similarity between the cells that we think they could be and the cells that you've actually given me. That's just an overview of the procedure, more for the clinical audience. I'm not sure how many clinical people are here, so I'm not going to go through it. But a recap, high parameter analysis, there's a reason to do it, improves its accuracy, enables discovery of new features. But in order to make use of that, you really do have to develop new informatics approaches in order to look at the data in an efficient manner or interpretable manner, and to apply it clinically. What's the point? Well, we discovered new subtypes of B cells that determine whether you're going to need chemotherapy or not. And maybe that's something important to know. Can that inform how we manage patients? I hope so. And also just by doing research, it's kind of like the motivation for exploring space or having a big defense budget. There's trickle down benefits to other things, and so the trickle down of the methodologic and informatics from the kind of the research questions has really kind of, I think, increased the accuracy and sufficiency of our clinical efforts. So with that, I'll just acknowledge the people who did all the work. Shu Hai led and started off all the high parameter work on the follicular lymphoma with Michael's help. Then Michael kind of transitioned to the DLBCL work. Shu Hai is still helping, but now Michael was leading that one, and Ainsley has done all that kind of programming stuff I talked about at the end. There's lots of people who actually make all this happen, the research core, the clinical labs, and the funders. So with that, I guess I'll take any questions.
Thanks very much, Andrew. Is there any questions? You can either put them in the chat. Oh, okay. Weng?
Oh, hi, Andy. Excellent job and excellent talk. I just sorry I missed the beginning of the talk. You already covered my question, but I just want to ask, what's the current landscape of the flow cytometry surveys in the province, and what's your perspective of the future landscape and the role of BC Cancer Flow Lab in those landscape?
Sure. So that's a complicated question. It depends on how you want to deliver care in the province. There's kind of competing priorities here. Do we deliver care locally, and so that each area takes care of its own patients? I mean, there's something to be said for that. You need an intimate connection between the pathologists who serve the oncologists, and that's where the data is derived, and they're grown-up people, they can do their own work. But then there's also this issue of, well, how much can we really propagate the latest assays, to all the people in the province? People in smaller hospitals, other regions, are they getting less care? That's a much more complicated question, but that's not to say, I think generally where people are headed is there has to be a tiered approach. Look, if you live in an outlying area, you're not going to get the latest, greatest stuff. But if you need it, not everyone does, but if you need it, can you access that? And then you generally have to go to a center. So this kind of notion of delivery of care for the masses at a kind of a lower, sophisticated assay. But for those people who need it, then they could get referred into centers, cancer centers for more nuanced care, if you will. But it's always going to be, you can't deliver the highest level technology to everyone in the province. So there has to be some sort of compromise, some way to triage people who need more information gathered or people who need less information gathered. The one thing I'll comment is high parameter flow, it doesn't cost any more to do the high parameter flow. I mean, if you think about the real cost of processing patient samples, it's all the human stuff. Dropping another 1 to 15 antibodies in a tube, that's really not, and the cost of antibodies is trivial. That's really not the issue here. But I think I don't think I skipped over that part in the slide because I didn't think there were many clinical people listening. But nonetheless, some of these tools we've developed intentionally with a mind that it's not just for our platform, for our panel. These methodologies that Ainsley was working on, we've specifically tried to design them so that if you have another panel, you have another platform, it doesn't matter. These informatics approach are agnostic to that. So I'm hoping that in that way, some of the stuff that we are learning, we can help other sites analyze their data. Because at some point, they're not going to have the human capacity to do it. It's just too slow and inefficient, non-reproducible. So anyways, there are some things we can deliver. I think informatics is something that should be able to transcend these geographical boundaries.
Thank you.
Jennifer?
Yeah, thank you, Dr. Weng. I was just wondering, I noticed in your presentation you use flow cytometry a lot for your diagnostic work or detection system. Have you ever used multiplex immunohistochemical staining techniques instead, or is there some reason why you didn't use them in this particular presentation?
So multiplex IHC practically is limited to about 3 to 5 colors, and it's the analysis of that data. It's kind of more direct, and if you're a pathologist, you want to look at the tissue, you want to look at the cells. We've looked at high parameter image, in situ imaging mass cytometry. We tried that. We tried multiplex ion beam imaging, maybe we tried that. And now on a research side, they're doing CosMX, which is panel transcriptomic analysis on tissue base. So, it's a matter of complexity, but it's also a matter of how you can analyze data. Flow cytometry, by definition, is relatively easy to analyze with existing informatics tools because the data is basically transformed into a CSV file. It's just their data. It's a matrix of values, so it's actually quite amenable to the kind of the informatics tools we have at the moment. Multicolor IHC, CosMX, imaging mass cytometry, those all require segmentation, which is some computer has to go in there and circle and say, that's a cell. And this is where this cell ends and that one begins. I've seen a lot of the efforts that Christian Seidel and David Scott and their teams have been looking at. And I tell you, it's not an easy problem to solve. You might think it's easy because you're a human to see, oh, that stain is on that cell. I can tell the difference. It's not so easy for a computer. And so while we have experts and people that can do that visually, that's not scalable. So until we solve the segmentation issue, I think it's not going to come online. But if we can solve the segmentation, then yes, multicolor IHC could be scalable at a higher complexity clinically.
Thank you.
Great. Tyler?
Hey, Andy.
Hey.
Great work. It's neat to see how far things have come. Do you see, so obviously you're kind of using these kind of Shuhai's developed AI tools to kind of simplify the interpretation for the pathologist side. Do you see that continuing further in its sophistication as AI in general gets more powerful? I think there's never going to take away the human completely, but do you see that progressing further in the years to come?
Yeah, that's a great question. So actually, I apologize. I did skip over this issue in that slide I skipped over, but I guess maybe I can just go back to that just for have us to look at something while we talk. But I wanted to point out that I don't think, but informatics people can chime in. I don't think what we're doing is AI. AI is, my understanding is, the computer has to learn the patterns themselves. In fact, the AI is done with it. You have actually no idea what it was the AI what reasons the AI
Yeah.
had for telling you that's a picture of a cat versus
Yeah.
that's a picture of a dog. You don't know what the features are. You just have to trust the computer. And that, for all those of you who have, used AI search tools, you get some crazy answers. You're like, I don't know. So a
Yeah,
lot of
I think some of the models are starting to be more transparent. I
yeah,
think, but
right.
like the Chinese version, I think
Yeah,
that was the appeal of it, is you could actually
yeah.
get some information that way. But I agree, it is sometimes a bit of a black box.
Yeah. So what we're doing here, though, it's not AI at all. I don't think it's machine assisted, but it's not machine learning.
Yeah.
We're basically telling the computer what to do, but in an automated fashion. So we annotate, we use ground truth. Now, every AI has to have annotated data files, fine. Of course, AI needs thousands or tens of thousands, and we've only done this with a couple hundred so far. Multidimensional clustering, that's just looking at data in multi-dimensions. I don't think that's AI.
So you're
Collapsing
just training it how to analyze and segregate the data, basically.
Yeah, I mean, essentially, we're just saying if you drop all your marbles on the floor and then form them into piles, and then you say, well, this pile is that, and this pile is that, and then you come in with another cup of marbles and you dump it, and you say, where do those marbles fall? Well, some of your marbles fell into a bucket that I know tends to always be containing CLL cells. Another bucket of marbles fell into, well, those are normal B cells. All we're really doing is asking the computer to take another unknown sample and tell me, where do those marbles fall? They're cells, sorry, not marbles. Where do those cells fall? Have we seen these before? Are they like cells we've seen before? And if they look like cells we've seen before in multi-dimensions, what were those cells what did we call them the last time? So it's really just pattern matching, but a computer is doing it in multi-dimensions for you. It's not learning anything, it's just doing what we do, but in a more high dimensional and reproducible fashion. That's not AI in my mind, but
Yeah.
and I think the issue is you get to make the decision. All we're saying is these cells fell into this bucket.
Yeah. And
I think they're CLL. Do you think they're CLL?
yeah.
Right. So
No, more, I guess, given that there's been some publications now about radiologists versus AI and stuff like that. And yeah, you don't know what's going on in the black box, but it seems like it might be not a bad thing to have some kind of automated, even if you don't know how it's doing it, its own interpretation of things as a check on the pathologist. So you can almost have like, if there's something that it highlights that the pathologist didn't highlight, it might make that person just, oh, okay, maybe I should just make sure there's nothing there. Not to say that you're going to let it do the sign out, but it's more just like as a way of like an extra layer of kind of
right,
check, I guess.
more of an augmented reality than necessarily
Yeah.
actually giving the
Almost
card.
like having a second pathologist verify
Yeah,
what you're
yeah.
signing out.
I have to admit that when we first started on this idea, Ainsley's first inclination was to use a neural network, like a genuine AI tool. It didn't go so well. So, not to say that things couldn't go better in the future, but I think we're using the tools that we trust at the moment, and I think we can still help in the near term, the clinical workload we're doing. Yeah, maybe AI will catch up and then we'll trust it. But until then, this is what we're doing.
Yeah, I wouldn't want to rely on it completely though.
Not yet.
Thanks.
Yep.
Okay, are there any other questions? If not, we're right on time, which is pretty amazing for us. So if not, thanks very much, Andrew. That was an awesome talk, and I really
Will.
appreciate you taking the time to.
If anyone has any questions, feel free to contact me. I'd love to chat about this stuff more.
Fantastic. Thank you very much.
Thank you.
Have a good weekend, everyone.